I'm here to discuss CNVScope, which is an R package and Shiny application that looks at copy number relationships to implicate genomic regions um, in cancer driving events. To give a bit of background, changes in copy numbers can occur in disease processes, including amplification, duplication, and translocation events. Um, Amplifications can be illustrated here, where there's an increase in the number of copies um, of on a chromosome or external to a chromosome. Deletions can occur where there's a decrease in the number of copies of a chromosome. Translocations are where portions of DNA switch places. Um, this can happen with or without copy number change. Um, and and one of the most famous translocations is the 922 translocation in chronic myeloid leukemia, which creates the Philadelphia chromosome, which creates the BCR-ABL fusion protein. And this discovery of discovering the translocation, which led to this discovery of the protein that drove the cancer, allowed for a drug called imatinib or Gleevec to be created, which inhibits the tyrosine kinase and stops CML, stops the cancer. And so it was a big deal. It was on Time magazine, and it was the award-winning discovery that was featured in Cancer of Emperor Maladies, which is a Pulitzer Prize-winning book. It really transformed leukemia treatment and cancer research. And so we feel that using numerical measures of copy number, we may be able to understand the cancer better and lead to new treatments. But without understanding the mechanism of translocation, such a therapy would not have happened. So one more vocabulary piece to know is double minutes, which are circular DNA external to the chromosome. Typical copy number representations are given in terms of changes of gains and losses across the genome. And you can see the same thing here, where you have the big red peaks designate amplifications, and the blue downward peaks are deletions. But typically, it's not thought of to look at the relationships between these peaks. Does a deletion here associate with an amplification there? That question at the time hadn't really been asked in the literature much, and so typically, and not at a grand scale that involved looking at regions of association or domains. And so that's the key idea of CNVScope, that we look at regions of association between copy number changes. So in order to do that, we're going to create Hi-C-like plots with domains found using Hi-C two-dimensional edge finding or segmentation algorithms. Another type of plot are Circos plots. These are really pretty, but as we'll discover, CNVScope is able to be a bit more easily interpretable. And so to give a little bit of background on Hi-C, it's a technique that measures the contact frequency between DNA strands, which allows the application of loops or domains in DNA. And so each point in this matrix is a measure of contact frequency, where one region is associated with another region. and by using segmentation algorithms, they're able to find these domains, these squares, which implicate loops where regions of DNA are proximal and associated. Similar to this, CNVScope finds domains where DNA is associated. And so what's really important about this is we see two domains which correspond to two arms of a chromosome. Chromosomes have typically have two segments, a P and a Q arm, and these breaks in correlation correspond to the centromeric region, this place where the P and the Q arm touch. So essentially, numerical copy number association with neighboring DNA suggests that in neuroblastoma, this DNA typically remains connected. So the arm remains connected with the rest. If the arms were heavily segmented into smaller domains, we might expect some event of some kind, as we'll see. This is how the data was processed. Without running out of time, essentially the matrix is made by linear regression. The negative log of the p-value is multiplied by the sign of the Pearson correlation. This is done in a highly parallelized fashion, where you can break this apart into thousands of jobs and get this done in about half an hour. It's really scalable. We also designed a custom metric to make the color scaling work, which you may not appreciate, but it was a fine an endeavor which took a bit of fine tuning. Additional measures of association are implemented, including Pearson, Spearman, and Kendall correlation. And you can find that in the additional examples vignette on CRAN. We use several segmentation algorithms. We tried image segmentation and many different types, but ultimately we found that three algorithms work the best. We include Hi-C-Seg and BlockSeg, which Hi-C-Seg is best for symmetric chromosomes, like chromosome 11 and 11. BlockSeg is great for asymmetric chromosomal pairs, so chromosome 1 and 2, for instance, or chromosome 17 and 19. In order to choose, so essentially, in order to choose these regions of association, we found that joint seg actually did the best, and was able to pick out these regions of association. If you notice, these are the places labeled designate the segmentation boundaries. And so these domains denote association of the number of copies of DNA, which can be predictive of their physical association or dissociation. The number of chromosome arms, for example, chromosome arms are typically associated, which form a domain for each arm. So the app provides a whole genome view, and typically a view of a pair of chromosomes. And you can search for genes. You can look at and zoom in and find genes within a certain clicked region. You can also find which genes are located in the cancer gene census and determine what cancers these genes are typically involved in and what their mechanism, if known, is. Further, you can drill down and really get at the granular single sample copy number data and find out where these associations coming from. Are they driven by a single sample, or are they, a general feature of the data that happens across multiple individuals? One thing to note here, this region that's blue here designates a loss and gain association. And in the paper, we're very careful to just say that there's a loss association and there's a gain association, and this is known to occur in neuroblastoma. Subsequently, I found that in a recent publication, an 1117 translocation has been characterized. But anyway, there's a whole tutorial online where I go through each piece of this. CNVScope is built to handle to highlight copy number drivers in cancer. And the main figure shows a break in the typical correlation structure on chromosome 2. If you'll notice, there are these white lines emanating directly from NMIC, which has been suggested to be the best characterized genetic marker of risk in neuroblastoma. So CNVScope not only confirms what's known to be cancer associated, it highlights NMIC, which is a cancer associated gene, but the pattern aligns with the mechanism, the double minute mechanism in the cancer, where there's this dissociation of DNA of copy number association is associated with the dissociation of the physical DNA from the chromosome. And so the DNA becomes associated not with the chromosome, but with these double minute DNA circles, extra chromosomal DNA. And after we published this, there was a publication in Nature Genetics that supported this and showed the double minute mechanism as well, and supported this this mechanism that CNVScope shows. This package is available on CRAN. We have lots of vignettes that allow the user to construct their own matrices from scratch using TCA or target data from beginning to end. I'm very open and transparent about how this is done. There are additional examples, which I won't get into here. You're free to look at them. But in summary, so future directions, we've found that there are interesting events on some of our internal data. We're working on it. I can't say more than that. So please don't ask, but it'll be worked into a future paper. So we're continuing to explore and find the meaning of these associations. In summary, CNVScope is a statistical framework to visually model copy number associations in order to understand physical DNA associations in cancer, in order to understand breakage events that trigger oncogenic copy number change in cancers and relate them to known cancer drivers. The aim is to initiate new clinical trials based on inhibitors of the genes associated with copy number change. I've included references, and thanks to the many wonderful people I've worked with and corresponded with at the NIH and outside the NIH. Thank you for listening.